Dear students, welcome to the second part of this lecture on neuromotor control. The first part looked at the brain structures, especially the brain structures involved in the control of voluntary movement. And then now we're more going to look at alternative structures involved in neuromotor control pathways, and then finally some of the effects in which we can see the central nervous system at work. So first of all, as you can see here, there's a neuron or a nerve cell in this particular picture. A nerve cell is between 4 and 100 microns large on average, right? 100 microns is only 0.1 millimeters. But of course, there are some nerve cells that are far longer. There are some nerve cells that have axons that are about 1 meter long. Right? And neurons, as you can see here, they've got specialized projections, and those projections are either axons or they are dendrites. Dendrites take information from other cells and move that to the cell body, while axons take information away from the cell body to other cells. There are about 86 billion neurons in the brain. When we look at the types of neurons that we have in the human body, there are sensory, motor, and interneurons. Sensory neurons are also sometimes referred to as afferent neurons. They are called afferent neurons or ascending neurons because they take information from sensory receptors, which we sometimes refer to as the periphery. They take information from sensory receptors to the central nervous system. On the other hand, we've got motor neurons, which are referred to as efferent neurons or descending neurons. And they take information from the central nervous system to the skeletal muscle, which are often commands to contract skeletal muscle. And then finally, there's interneurons or relay neurons, which originate and terminate in the brain or spinal cord and connect motor and sensory neurons. There are different neural pathways, of course, depending on whether we talk about bringing sensory information to the central nervous system or taking motor commands from the central nervous system to the periphery. We either talk about sensory neural pathways or motor neural pathways. Sensory neural pathways are called ascending tracts, and they pass through the spinal cord and the brainstem and then connect to the sensory areas of the cortex of the cerebrum, the brainstem, and the cerebellum. Motor neural pathways work in the other direction. They're descending tracts. They go from the brain through the spinal cord and end up in the periphery. And there's two types of tracts. First, there's the pyramidal tracts. These pyramidal tracts directly innervate motor neurons of the spinal cord. And then there's non-pyramidal or extrapyramidal tracts. And they are mainly involved in involuntary actions. They are mainly found in the pons and medulla oblongata, and they're involved in reflexes, locomotion, postural control, etc. So pyramidal tracts are more involved in motor functions, voluntary motor functions within the body. Non-pyramidal tracts or the fibers that run through there, they are involved in involuntary motor control. And as you can see on this picture, you can see all the efferent or descending pathways in red, and you can see all the sensory or ascending pathways in blue. And then you can see that only in the motor or the descending, the efferent pathways, do we have pyramidal tracts and we have extrapyramidal tracts, depending on whether or not fibers run through there that are involved in either voluntary or involuntary control. Then in the top picture here, you can see what a sensory neuron and a motor neuron actually looks like. So if we start at the top, let's say that we have temperature receptors in the skin, right? We then transmit that sensory information through the axon of the sensory neuron. That then gets communicated through an interneuron and ends up in a motor neuron. And that motor neuron then contracts skeletal muscle to move our hand away from, for example, a very hot surface. And the way in which cells communicate is through something called a synapse, and you see them in the bottom picture there. So a synapse is a structure that permits one nerve cell to communicate with another by passing an electrical or a chemical signal. That happens when one cell is in proximity to another cell, and we call the first cell, we call that the synaptic cell, and the second cell, the target cell, we call that the postsynaptic cell. So often the presynaptic cell is the axon, or the presynapse is from the axon of one cell, and the postsynaptic part is the dendrite of another cell. So this is how cells communicate. And what happens is a neurotransmitter gets released, a chemical signal that then alters the electrical current or the electrical charge of some of the receptors and cells, etc. So this is a good way for for neurons to communicate with each other, despite the fact that there's maybe no real physical contact. And we call that lack of physical contact, or that gap between two cells, we call that the synaptic cleft. So the axon terminal of one cell communicates with the dendrite of the other cell, of the postsynaptic cell, through the synaptic cleft using a neurotransmitter. Now we'll talk about that more in your lab, specifically when we talk about Parkinson's disease. Then once information or once a motor command reaches the muscles, of course the muscle needs to get contracted. And that happens in a motor unit. And a motor unit consists of the alpha motor neuron and all of the fibers that it innervates. And sometimes a motor unit can innervate just a few muscle fibers, but sometimes a motor unit innervates a whole lot of muscle fibers, right? And there are as many as 200,000 motor units inside the spinal cord. And the number of muscle fibers that you can activate at once is a direct relation. There's a direct relationship with the amount of active motor units. Apologies. So how do we actually control voluntary movement? Well, there's actually 4 steps. Let's say there's 3 steps and then one additional bit of information. So you can see on the left picture that there are different levels of control hierarchy. So you can see here there are higher centers, then there's the cortex, basal nuclei, thalamus, brainstem, cerebellum, and then finally we move to the last, or the local level, brainstem and the spinal cord. So I'll try and talk you through these different levels. So the first step, to carry out goal-directed movements, the motor cortex must receive different information, or a variety of information, from all the different brain lobes. That's where it starts. And we've talked about the basal ganglia before, in this case basal nuclei. So the basal ganglia integrate all of this information, and once it's processed, they feed it back to the thalamus, which we previously described as a relay station. And that is how we select and inhibit coordinated movement voluntarily, which you will see later as well, that why the basal nuclei or the basal ganglia play such an important role in the mechanism of Parkinson's disease. Then eventually the cerebellum makes this movement smooth. So once we have inhibited or in this case selected movement, then the cerebellum makes the movement smooth, because the cerebellum precisely regulates the sequence and duration of each segment of a movement. So the cerebellum receives information about the intended movement from the sensory and the motor cortex, and then information is sent back to the motor cortex, so movement, force and precision, etc. can be adjusted. Now, this is the basic, or this is how we control or basically describe the hierarchy, because then motor neurons carry signals to muscle fibers. those muscle fibers contract muscles that then generate sensory information that we again use to adapt motor control, right? But of course, there's an activation sequence for motor areas as well, which you can see on the right-hand side. So the planning for every given movement, the highest center of control, right? The planning for every given movement starts in the frontal lobe. And the frontal lobe issues commands to a particular part of the motor cortex. It's here described as area 6, which you can see in the picture. And in area 6, we decide which muscles to contract. And then the command goes further to area 4 to finally activate specific muscles to achieve that movement. That information is then sent through the spinal cord to the periphery using motor units to contract skeletal muscles. And that then generates a feedback loop with sensory information. So by using the first 3 steps, right, until the cerebellum, plus then understanding the activation sequence for motor areas and understanding the reciprocal nature of the sensory and motor pathways, you can really understand how the role that the brain plays in controlling voluntary movement. Now, I find it important to not just talk about the dry aspects of voluntary control of movement and the role of the central nervous system, but it's also important that we discuss what some of the outward influences are that we can see of the central nervous system. So there are 3 ways in which we can see the external effect of deficiencies in the neuromotor system. We can see it in handedness. We can see it in how experts' brains are maybe structurally or functionally different from non-experts. And we can look at it in terms of neurological disorders or neurodegenerative disease. So first I want to talk about handedness. So the human brain is composed of 2 halves. We all know that. We talked about the cerebral hemispheres before. And a person's handedness is opposite from the specialized hemisphere for language. And it's called the division of labor explanation for handedness. So in this case, if we think back about 350,000 years ago or even more, right, when we were all hunter-gatherers, right, both speaking and handedness require really fine motor skills. Yeah, to move your lips to get really varied sound and to be able to do things like making flints and shaping rocks to be spear points, etc. Both of those require fine motor skills. So having one hemisphere that does both is much more efficient than dividing it up. Right? So that's the reason why we say that there are more right-handed people than there are left-handed people, because the right brain hemisphere is also important in fine motor control. Handedness is about 24% inheritable. Left-handedness is more common in men, and we can talk about why that is later, but there are some interesting explanations, but I might keep that for the Q&A. And just for your information, 4 of the last 7 presidents have been left-handers. Additionally as well is that left-handedness also exists in animals. For example, I looked this up, kangaroos show a preference for left-handedness. And however, true handedness for marsupials is very difficult to determine because they don't have a corpus callosum. So if you listen to the first lecture, you know why that is important in determining handedness. You might have heard of the theory of hemispheric dominance. So some tasks are related to a hemispheric dominance. For example, language skills such as grammar and vocabulary, etc., are lateralized to the left hemisphere. And that sprouted a theory that left-handed or right-brained people are more artistic and creative, while left-brained or left-brained, right-handed people are more analytical and methodical. But there is very limited empirical evidence that exists in support of a very simplistic theory like this of hemispheric dominance. That being said, there are some interesting studies that I found where people looked at the distribution of left-handedness and right-handedness in very creative populations, very aggressive populations. So if you're interested in handedness and its relationship with not just aspects of society, but perhaps also in sport, please let me know and I can share some information with you. Another way in which we see exceptional functionality or dysfunctionality of the brain, in this case functionality, is through the expert's brain. Now, learning has a massive effect on brain function and often on brain structure, right? And before you started listening to this lecture, and after, your brains are not the same. They have functionally changed, right? You have made new connections, etc. But there are some interesting studies that also look at structural brain changes. So whether the actual brain changes in its structure. And one of the most interesting studies, and I won't talk about this too much because it's part of your laboratory, is one on London taxi drivers who had to learn. Back then, you still had to learn all of the streets and all of the important points and the good nightclubs and the good restaurants, etc. by heart. And they looked at whether expert taxi drivers or those that succeeded in training, whether they had different brains in an fMRI scan, whether they had different brains than taxi drivers who weren't as expert. And they saw that the posterior hippocampus was enlarged in expert taxi drivers. And the posterior hippocampus is really important for things like spatial memory. So what that demonstrates to us is that structural brain changes can be achieved in some high-level experts. However, we are much more likely to see functional brain change. And functional brain change relates not to a change in the structure of the brain, because obviously the brain is captured by the skull, right? It can't keep growing forever, right? But what can change is the synaptic connections inside the brain, right? And I'm leaving that up to your lab and up to yourself, but that has warranted some discussions around brain plasticity in children versus adults, etc. Then finally, we're going to talk about some neurological disorders, but I'm going to keep this part really short because, again, it is part of your laboratory and part of some of the exercises that you'll be doing there within a group. So the first neurological disorder that we're going to look at is called Parkinson's disease. And most people would be familiar with Parkinson's disease, which is basically a disorder of the basal ganglia. And it has something to do with a lack of dopamine, right? And we know that dopamine is a neurotransmitter. And if you know what a neurotransmitter does in the communication between nerve cells, you should be able to figure out the basic mechanism behind Parkinson's disease. Now, Parkinson's disease is often recognized by slow movements, which is referred to as bradykinesia, reduced amounts of movement, tremor, which we all know, and muscular rigidity. Now, interestingly, there are some really advanced interventions that have been used to try and help Parkinsonian patients. So what happens to a Parkinsonian patient is that sometimes their ability to control voluntary movement gets overwhelmed in such a way that they freeze, right? They can't move whatsoever. And a lot of aged care facilities or specific Parkinson's patient hospitals, which I've been to a few of them in Brazil, which was very interesting, have found some really interesting and really advanced interventions to help these Parkinsonian patients to continue to move once they freeze. And one of the most interesting interventions they have is in Parkinson's hospitals in Brazil. There are lines painted on the floor so that Parkinsonian patients, when they freeze, they can focus on something external to their body, right? They can try to step over one of these painted lines on the floor, and that temporarily unfreezes them. So if you're interested in finding out more about how hospitals and how aged care facilities have tried to make the life of patients with Parkinson's disease more tolerable, you can look into some of those studies or just ask me. Then multiple sclerosis. Well, multiple sclerosis is a demyelinating disease. And when we talked about sensory neurons and motor neurons and interneurons before, what we would have seen is that around the axons of a neuron is a myelin sheath. And a myelin sheath is a fatty sheath that is wrapped around the axon, and it helps with signal transmission. But if that myelin sheath isn't there or it's damaged, then you get a lot of signal leakage. What that means is that if you send 100% of signal, only 20% might arrive at the end of the axon, right? So if you get a demyelinating disease where the myelin sheaths are damaged, sorry, that should be sheaths, not sheets, then obviously you get all kinds of externally visible symptoms, things like double vision, blindness in one eye, and troubles with coordination. And you can see the difference between a normal neuron and a neuron where the myelin sheath is damaged. And you can also see on the bottom there what that does to signal transmission, right? So you get a much less strong signal or electrical potential that moves through the neuron. And then finally, ALS, amyotrophic lateral sclerosis. Well, most well-known person with ALS is Stephen Hawking, obviously. And what ALS is it is a progressive degeneration of motor neurons. And in this case, the brain loses the ability to initiate and control movement, right? So we've all seen the role that the brain plays as a higher control or a higher level of control in the control of voluntary movement. And because of the fact that the brain loses the ability to control and initiate movement, muscles atrophy. And that gives obviously very, very pronounced difficulties, not just in walking, but eventually also in breathing, etc. So in summary, what did we do in this entire lecture? Well, first of all, we examined all of the features of neuromotor control, perhaps not in extreme detail, but I find it very important that you know just enough to understand what happens when someone has a neuromotor control system that doesn't operate effectively. And we've looked at that specifically afterwards, where we've investigated how neuromotor control influences motor behavior in healthy and unhealthy populations, where we've specifically looked at expert versus novice brains, and we've looked at handedness, and we've looked at neurodegenerative disease. Hopefully this was useful to you. Please remember to note down your questions for the Q&A session that is upcoming. All right.